Chapter number 1. Cell structure and function. Cell theory and magnification. 1. Schwann equals zoologist, Schleiden equals botanist. 2. Magnification equals apparent size of an object / actual size. 3. Compound microscope equals 10,000x equals a piece x objective. 4. Electron microscope equals 1,000,000x. Resolution. 5. To distinguish between two closely related objects being separate from one another. 6. Human eye equals 1 millimeter. 7. Compound microscope equals 250 nanometers 500x smaller in value. 8. SEM equals 3D image. Staining. 10. Counterstains are used with primary stains. 11. Counterstains are used to stain other tissues than main tissue. 12. Fungal hyphae and spore equals aniline blue. 13. Nuclei equals methylene blue / safranine / borax carmine. 14. Lignin equals Schultz solution / safranine / aniline sulfate. 15. Starch equals Schultz solution / iodine solution. 16. Cellulose equals Schultz solution / eosin. 17. Supernatant liquid, no PPT at certain speed. 18. Nucleus 800 G 10 minutes. 19. Mitochondria plastid, 20,000 G 15 minutes. 20. 100,000 G cell membrane microsomes 1.5 hours. 21. 150,000 G ribosomes 3 hours. Tissue culture. 22. Micropropagation. 23. Totipotent cell and explant taken. 24. Agar semi-solid and broth liquid. 25. Callus equals undifferentiated mass of cells. 26. Differentiation is brought about by hormones. 27. Multiple shoots auxins low cytokinin high ratio. 28. Root formation equals high auxins and cytokinin ratio. 29. Cryostorage equals extreme cold storage. 30. Inverted microscope equals stage is upside down. It was invented in 1850 by J. Lawrence Smith. Chromatography. 31. Mikhail Tsvet. Chromatography was first employed in Russia by the Italian-born scientist Mikhail Tsvet in 1900. He continued to work with chromatography in the first decade of the 20th century, primarily for the separation of plant pigments such as chlorophyll, carotenes, and xanthophylls. Since these components have different colors, green, orange, and yellow, respectively, they gave the technique its name. 32. The mixture is dissolved in a fluid called the mobile phase, which carries it through a structure holding another material called the stationary phase. The various constituents of the mixture travel at different speeds, causing them to separate. The separation is based on differential partitioning between the mobile and stationary phases. 33. The solvent moved by capillary action through paper. Capillary action, sometimes capillarity, capillary motion, or wicking, is the ability of a liquid to flow in narrow spaces without the assistance of, or even in opposition to, external forces like gravity. The effect can be seen in the drawing up of liquids between the hairs of a paintbrush, in a thin tube, in porous materials such as paper and plaster, in some non-porous materials such as sand and liquefied carbon fiber, or in a cell. It occurs because of intermolecular forces between the liquid and surrounding solid surfaces. If the diameter of the tube is sufficiently small, then the combination of surface tension, which is caused by cohesion within the liquid, and adhesive forces between the liquid and container wall act to propel the liquid. Electrophoresis. 34. The separation of molecules by different electrical charges. 35. Size/shape/ of molecule amount of charge. Spectrophotometry. 36. Every chemical compound absorbs, transmits, or reflects light, electromagnetic radiation, over a certain range of wavelength. Spectrophotometry is a measurement of how much a chemical substance absorbs or transmits. Spectrophotometry is widely used for quantitative analysis in various areas, example, chemistry, physics, biology, biochemistry, material and chemical engineering, clinical applications, industrial applications, etc. 37. A spectrophotometer is an instrument that measures the amount of photons, the intensity of light, absorbed after it passes through sample solution. With the spectrophotometer, the amount of a known chemical substance, concentrations, can also be determined by measuring the intensity of light detected. Depending on the range of wavelength of light source, it can be classified into two different types. UV visible spectrophotometer uses light over the ultraviolet range, 185 to 400 nanometers, and visible range, 400 to 700 nanometers, of electromagnetic radiation spectrum. IR spectrophotometer uses light over the infrared range, 700 to 15,000 nanometers, of electromagnetic radiation spectrum. In visible spectrophotometry, the absorption or the transmission of a certain substance can be determined by the observed color. For instance, a solution sample that absorbs light over all visible ranges, i.e., transmits none of visible wavelengths, appears black in theory. On the other hand, if all visible wavelengths are transmitted, i.e., absorbs nothing, the solution sample appears white. If a solution sample absorbs red light, 700 nm, it appears green because green is the complementary color of red. Visible spectrophotometers, in practice, use a prism to narrow down a certain range of wavelength, to filter out other wavelengths, so that the particular beam of light is passed through a solution sample. Microdissection. 38. Dissection under microscope. 39. Ocular equals non-calibrated micrometer. 40. Stage micrometer calibrated. Cell wall. 41. Non-living. 42. Permeable membrane. 43. Apoplast movement equals movement through cell wall. 44. Secreted by A. Golgi bodies B. Protoplast. 45. Polysaccharides. 46. Homopolysaccharide cellulose optically active. 47. Heteropolysaccharides pectine/hemicellulose/lignin. 48. Middle lamella optically inactive/pictinate is a structural heteropolysaccharide contained in the primary cell walls of terrestrial plants. It was first isolated and described in 1825 by Henri Braconnot. It is produced commercially as a white to light brown powder, mainly extracted from citrus fruits, and is used in food as a gelling agent, particularly in jams and jellies. 49. Primary cell wall equals optically active/matrix/cellulose, hemicellulose equals 2:1, optical rotation or optical activity, sometimes referred to as rotary polarization, is the rotation of the plane of polarization of linearly polarized light as it travels through certain materials. Optical activity occurs only in chiral materials, those lacking microscopic mirror symmetry. 50. Secondary cell wall equals sclerenchyma/lignin/optically active. Lignin is an organic substance binding the cells, fibers, and vessels which constitute wood and the lignified elements of plants, as in straw. After cellulose, it is the most abundant renewable carbon source on Earth. Between 40 and 50 million tons per annum are produced worldwide as a mostly non-commercialized waste product. It is not possible to define the precise structure of lignin as a chemical molecule. All lignins show a certain variation in their chemical composition. However, the definition common to all is a dendritic network polymer of phenylpropene basic units. There are two principal categories of lignin, those which are sulfur-bearing and those which are sulfur-free. It is the sulfur-bearing lignins which have to date been commercialized. These include lignosulfonates, world annual production of 500,000 tons, and kraft lignins, under 100,000 tons pa. Due to the lack of suitable industrial processes, the sulfur-free lignins are as yet non-commercialized. 51. Nitrocellulose equals gun cotton. In 1862, the first man-made plastic, nitrocellulose, branded Parksine, was created by Alexander Parks from cellulose treated with nitric acid and a solvent. In 1868, American inventor John Wesley Hyatt developed a plastic material he named celluloid, improving on Parks' invention by plasticizing the nitrocellulose with camphor so that it could be processed into finished form and used as a photographic film. 52. Rayon textile fiber. Cell membrane. 53. In prokaryote cell division/DNA replication/respiration/mesosome formation/photosynthesis/reverse etc equals NADPH synthesis. 54. Lysosome add to the surface of plasma membrane. 55. Golgi bodies form plasma membrane. 56. Integral proteins / intrinsic proteins H+ pumps / transporters / facilitators. 57. Facilitated transport equals movement through plasma membrane proteins. 58. Lipid bilayer allow nonpolar and prevent ionic materials / differentially permeable. 59. Membrane protein make PM selectively permeable / select material according to cell need. 60. Extrinsic proteins act as a receptor, receive stimulus. 61. Intrinsic proteins regulate diffusion / osmosis / active transport. 62. HRS equals glycolipids / glycoproteins. There are two main classes of hormone receptors. Receptors for peptide hormones tend to be cell surface receptors built into the plasma membrane of cells and are thus referred to as transmembrane receptors. An example of this is insulin. Receptors for steroid hormones are usually found within the cytoplasm and are referred to as intracellular or nuclear receptors, such as testosterone, and nerve impulse recognition of antigen. 63. Membrane carbohydrates are responsible for endocytosis. 64. Glycolipids and glycoproteins act as a cell surface markers. Chronological points. 65. 1925, Gardner and Grendel, lipid bilayer only. 66. 1935, J.F. Daniell and Davison, lipid bilayer plus proteins and proteins pores. 67. 1959, Robertson, unite membrane model. 68. 1972, Singer and Nicholson, fluid mosaic model. 69. PM produce ionic gradient for nervous and muscular activity. Cytoplasm. 70. Cytosol sol form plants / endoplasm / thin. 71. Cytosol gel form animal / ectoplasm / thick. E.R. 72. Cisternae art.E.R. 73. Tubules S.E.R. Sarcoplasmic reticulum. 74. Nuclear membrane is formed from R.E.R. 75. AS.E.R equals Ca concentration / muscles contraction / detoxification / lipid synthesis / hormone synthesis lipid origin / Na regulation, aldosterone. 76. ER add vesicles to CIS Golgi. Ribosomes. 77. Pallotti 1955. 78. 50% rRNA 50% protein / equal amount of both. 79. rRNA enzymatic action. 80. Occurrence equals mitochondria / plastid / nucleus / polysome / art.E.R free. 81. Prokaryotic ribosome equals 70s equals 50s plus 30s. The Svedberg is actually a measure of time. It is defined as exactly 10 to 13 seconds. 100 fs equals a substance with a sedimentation coefficient of 26s. 26 times 10 minus 13s will travel at 26 micrometers per second. 26 times 10 minus 6 m/s. Under the influence of an acceleration of 1,000,000 gravities, 107 meters per square second, equals the Svedberg is the most important measure used to distinguish ribosomes. Ribosomes are composed of two complex subunits, each of which includes rRNA and protein components. In prokaryotes, bacteria, the subunits are named 30s and 50s for their size in Svedberg units. These subunits are made up of three forms of rRNA, 16s, 23s, and 5s for bacterial ribosomes. Ultracentrifugation yields intact ribosomes, 70s, as well as separated ribosomal subunits, the large subunit, 50s, and the small subunit, 30s. Within cells, ribosomes normally exist as a mixture of joined and separate subunits. The largest particles, whole ribosomes, sediment near the bottom of the tube, whereas the smaller particles, separate 50s and 30s subunits, appear in upper fractions. 82. Eukaryotes equals 60s plus 40s. 83. Ribosomes are formed by nucleolus. 84. ALSU plus SSU requires Mg++. 85. Hemoglobin in RBC is synthesized by free RBC. Golgi bodies. 86. Camillo Golgi, 1898. 87. Cell secretion. 88. Concerned with glycosylation equals addition of polysaccharides. 89. Sulfation equals addition of sulfur. 90. Cargo of cells import-export things. 91. Enzymes modifications. 92. Glycolipids and glycoproteins are formed inside Gb. 93. Cell wall formation. 94. Dictyosomes in plants, formation of cell wall. The dictyosomes forms a structure called phragmoplast between the dividing plant cells. Phragmoplast form new cell wall between dividing cell. 95. Cisternae. 96. Phosphorylation. 97. Packaging/add to the surface of cell membrane. 98. Gb part cis Golgi medial Golgi trans Golgi. Lysosomes. 99. Lysosomes discovered by de Duva in 1949. 100. Cellular digestion. 101. Surrounded by single membrane. 102. Contain hydrolytic and acid phosphatase enzymes. Acid phosphatase, ACP, EC number 3.1.3.2, is an enzyme which removes phosphate from other molecules during digestion. It catalyzes the conversion of orthophosphoric monoester and H2O to alcohol and phosphoric acid. The enzyme is most effective in acidic environment. 103. Primary lysosome equals the processed enzymes bud off as Golgi vesicle from trans Golgi. 104. Secondary lysosomes equals secondary lysosome are formed by the fusion of primary lysosome with phagosomes. They contain engulfed materials and enzymes. The materials are progressively digested by the enzymes. So it is also otherwise called as digestive vacuole. 105. Tertiary lysosomes equals once the contents are degraded it becomes a tertiary lysosome. Sometimes you get tertiary lysosomes left over when the contents can't be fully destroyed. 106. Breakdown lipids/carbohydrates/nucleic acid equals pH equals 5. 107. 50 diseases related to its malfunctioning I.G. Tay-Sachs and glycogenesis type 2. 108. Tay-Sachs disease. Tay-Sachs disease is caused by a genetic mutation in the hexa genes on chromosome 15. It is inherited from a person's parents in an autosomal recessive manner. The mutation results in problems with an enzyme called beta-hexasaminidase A which results in the buildup of the toxin GM2 ganglioside within cells. 109. Glycogenesis type 2 equals glycogen storage disease type 2, also called Pompe disease, is an autosomal recessive metabolic disorder, 1, which damages muscle and nerve cells throughout the body. It is caused by an accumulation of glycogen in the lysosome due to deficiency of the lysosomal acid alpha-glucosidase enzyme. It is the only glycogen storage disease with a defect in lysosomal metabolism, and the first glycogen storage disease to be identified, in 1932 by the Dutch pathologist J. C. Pompe. Peroxisomes. 110, H2O2 ______H2O. 111, peroxidase C3 plants _____ photorespiration. 112, glycolate to glycine. Glyoxysomes. 113, conversion of lipids to carbohydrates. 114, only in plants/oil-rich seeds. 115, glycolic acid oxidase/catalase. 116, known as microsomes/microbodies. Cytoskeleton. 117, Koltsov 1928 suggested. 118, Cohen 1977 confirmed. 119, present only in eukaryotic cell. 120, tubulin/actin/myosin/troponin equals proteins. 121, microtubules form cilia/flagella/basal bodies/centrioles. Cyclosis cytoplasmic streaming equals the movement of cytoplasm around the nucleus is due to microfilament. 122, microtubules/spindle fibers/cell division. 123, intermediate filament maintains cell shape. Centrioles. 124, they are small pair of cylinders outside nucleus. 125, 0.3-0.5 long and 0.2 m in diameter. 126, centrosome equals centrioles plus PCM peri-central material. 127, centrosomes are mTOC microtubules organizing center. 128, centrioles are absent in higher plants. 129, cell division and formation of cilia and flagella. Cilia and flagella. 130, axon M plus membrane. 131, 9 doublets plus 2 singlet. Mitochondria. 132, absent in RBC. 133, 1850 first seen in muscle cells. 134, mitoplast equals mitochondria without outer membrane. 135, self-replicating/circular DNA/ribosomes. 136, Christie enfolding/etc equals oxidative phosphorylation. 137, YEF1 particles/ATP synthase equals knob-like structure. 138, matrix equals Krebs cycle/linked reaction/fatty acid metabolism. 139, oxidative phosphorylation occurs inside inner membrane of mitochondria. 140, H+ are stored in intermembrane space equals chemiosmosis. Plastids. 141, self-replicating/proplastids. 142, leucoplast equals storing plastid. 143, amyloplast equals starch-storing leucoplast. 144, proteinoplast equals protein-storing leucoplast. 145, eleoplast equals oil/fat-storing leucoplast. 146, chromoplast equals plastids other than green. 147, rhodoplast equals red chromoplast in red algae. 148, pheplast equals brown chromoplast in brown algae. 149, chromoplast equals petals/ripe fruits/autumn leaves. 150, contain chlorophyll which resemble hemoglobin but contain Mg++. 151, chloroplast equals contain A, envelope B, stroma C, thylakoid, grana. 152, 50 or more thylakoids per grana. 153, PS1 outside thylakoid membrane. 154, PSI present inside thylakoid membrane. 155, intergranum non-green. Nucleus. 156, 10 m in diameter. 157, outer membrane continuous with R.E.R. 158, nucleolus outer granular/inner fibrillar equals form ribosome. 159, nucleolus attached with secretary constriction on chromosome. 160, nucleoplasm is matrix. 161, chromatids are attached at kinetochore/centromere. 162, onion 16/chimps potato tobacco equals 48/frog 26/drosophila 8 garden p14/ pigeon 80 human 46. 163, haploid gamete number/diploid equals somatic cell number. 164, monoploid in case of polyploidy. Previous ETEA questions from chapter 1st. 1. Number of chromosome in tobacco is equals 2009/49. At 40, 45b, 48c, 46d, 47. 2. Cellulose is the building material of equals 2013/47. F. Middle lamella B, secondary cell wall C, cell membrane D, primary cell wall. 3. A special protein carrier in plasma membrane is known as equals 2014/173. A. Catalase B, lipase C, permeases D, arginase. 4. Nucleus was discovered by 2013/171. A. Waldier B, T. H. Morgan C, Kohler D, Robert Brown. 5. Potato plastids which store starch are known as equals 2013/181. At Paramelon B, leucoplast C, glycoplasts D, amyloplast. 6. Microvilli are also known as equals 2013/147. A. Leaf veins B, Christie C, leaf midrib D, leaf midrib. 7. S.E.R makes equals 2012/59. A. Enzymes B, lipids C, proteins D, sugars. Chapter number 2. Biological molecules. Introduction. 1. Biochemistry, structure composition/function of biological molecules. 2. Water is the most abundant of all compounds. 3. Protein is the most abundant of all organic compounds. 4. Water, 70%/proteins 15-18%/carbs, 3:4%/. Lipids 2:3%/DNA, 1:0.25%/RNA, 6:1.1% enzymes plus hormones 2%/inorganic salts, 1%. equals 60 to 80%. 6. The functional groups add to diversity of organic compounds. 7. Condensation and hydrolysis definitions important. Water has to absorb 4.184 joules of heat for the temperature of 1 gram of water to increase 1 degree Celsius, degree C. For comparison's sake, it only takes 0.385 joules of heat to raise 1 gram of copper 1 degree Celsius. 8. The living bodies contain 70 to 90%. 9. Protoplasm cannot survive below 10% water. 10. Oxygen is electronegative and H is electropositive. 11. H bonding is weaker than covalent bond. 12. Water is universal solvent. Water dissolves all minerals in soil. 13. The force between water molecules is cohesive force equals glue force. 14. Adhesive force, the attraction of water to polar cellulose equals sticky force. 15. High specific heat, the amount of heat energy required to raise the temperature of 1 gram of water by 1 degree centigrade. 16. Water absorb large quantity of heat without increase in temperature. 17. High heat of vaporization, heat required to convert 1 gm 100 seawater into 100 sea vapors. 18. Without H bonds water boiling point is -80 C and freezing point is -100 C. 19. Water is most heavy at 4 C. 20. Water has a high surface tension equals thin film of water cover membrane / moist. 21. Water ionization help in pH maintenance. Carbohydrates. 22. Polyhydroxyaldehydes or ketones / substances which yield such compounds. 23. One carbon contain aldehyde / ketone and 2 or more hydroxyl group. 24. Most abundant biological molecule in nature. Monosaccharides. 25. Not hydrolyzed into more simple units. 26. Easily soluble in water. 28. Vindenivos / triose / tetrose / pentose / etc. 29. Trioses are the simplest monosaccharide equals glyceraldehyde. 30. Ribose and glucose is aldose sugar and ribulose and fructose is keto sugar. 31. Isomers, same molecular formula but different structural formula. 32. Ring forms, furans by ribose / deoxyribose / fructose. 33. Pyrene rings by glucose and galactose. 34. Monosaccharides are white crystalline powders. 35. Stereoisomers, compounds having different spatial arrangements. 36. Optical isomerism is stereoisomerism. Oligosaccharides. 37. 2 to 10 monosaccharides. 38. Glycosidic bond. It is a type of covalent bond that joins a carbohydrate, sugar, molecule to another group, which may or may not be another carbohydrate. 39. Disaccharides, the oligosaccharide producing 2 simple sugars on hydrolysis. 40. Sucrose equals glucose plus fructose / maltose equals glucose plus glucose / lactose equals glucose plus galactose. 41. Sucrose is sugar cane sugar / lactose equals milk sugar / maltose equals fruit sugar. 42. Maltose is formed in human digestive system as a result of starch digestion. Polysaccharides. 43. More than 10 monomers. 44. Tasteless and soluble in water. 45. Starch / glycogen / cellulose / chitin / hemicellulose / lignin / pectin. 46. 2 types of starch equals branched / amylopectin and unbranched equals amylose. 47. Glycogen animal storing material extensively branched. 48. Cellulose the most abundant carbohydrates / plant cell wall. 49. Cellulose cannot be digested by humans. 50. Cotton fiber is cellulose. 51. Chitin equals fungi cell wall / arthropod shells / NH2 containing / indigestible. 52. Human blood 100 milliliters contain 100 milligrams of glucose. 53. Grapes contain 27% of glucose. 54. Honey contain large amount of glucose and fructose. 55. Left-handed sugar is indigestible and right-handed amino acids are indigestible except bacteria which can use right-handed amino acids. Proteins. 56. Most abundant molecules in cell. 57. Amino acids equals 20 types. 58. Alpha carbon attached with 4 different groups. 59. R group is variable. 60. Nonpolar aliphatic R group equals methionine/isoleucine/glycine/alanine/valine. 61. Leucine/isoleucine and valine are branched-chain amino acids of the nonpolar aliphatic R. 62. Sickle cell anemia equals glutamate is replaced by valine. 63. Polar uncharged R group equals serine/threonine/cysteine/glutamine/asparagine/proline. 64. Aromatic R group equals tryptophan/tyrosine/phenylalanine, PKU disease. 65. Phenylalanine is a precursor for tyrosine, the monoamine neurotransmitters dopamine, norepinephrine, noradrenaline, and epinephrine, adrenaline, and the skin pigment melanin. Phenylalanine is found naturally in the breast milk of mammals. It is used in the manufacture of food and drink products and sold as a nutritional supplement for its reputed analgesic and antidepressant effects. 66. Positively charged R group equals histidine/arginine/lysine. 67. Negatively charged R group equals aspartate/glutamate/. 68. CN linkage equals peptide bond AA equals dipeptide bond/AA tripeptide bond. 69. Hemoglobin is formed of 4 polypeptide chain 2 alpha and 2 beta. 70. Alpha chain equals 141 AA/beta chain equals 146 AA/total 574 AA and 570 peptide bonds. 71. Insulin molecule equals 51 AA/alpha chain 21 AA and beta chain equals 30 AA/total 49 peptide bond. 72. In sickle cell anemia glutamic acid is replaced by valine. Fibrous proteins. 73. One or more polypeptide chains. 74. Water insoluble/some sheet-like. 75. Keratin equals hair/nails/fur/outer skin/. 76. Myosin in muscles. 77. Collagen in skin/ligament/tendon/bones and cornea. Globular proteins. 78. Folded polypeptide chains. 79. Water-soluble. 80. Hemoglobin/albumin of egg white/enzymes/antibodies/proteins of cell membrane. Levels of structure. 81. Sanger 1951 was the first person who determined the sequence of amino acid in insulin. 82. Primary structure, linear sequence of amino acids. 83. Secondary structure, spirally polypeptide chain. 84. Tertiary structure, 3D enfolded polypeptide chain. 85. Quaternary structure, 2 or more polypeptide chains in a large size molecule e.g. hemoglobin and insulin. Functions of proteins. 86. Building material of cellular membranes called lipoproteins. 87. Stored in seeds e.g. beans, pulses p. Lipids. 88. C/H/O/N/P present in lipids. 89. Non-polar/insoluble in water. 90. Lesser oxygen than carbohydrates. Acylglycerol, glycerides, more correctly known as acylglycerols, are esters formed from glycerol and fatty acids. Glycerol has 3 hydroxyl functional groups, which can be esterified with 1, 2, or 3 fatty acids to form monoglycerides, diglycerides, and triglycerides. Vegetable oils and animal fats contain mostly triglycerides, but are broken down by natural enzymes, lipases, into mono and diglycerides and free fatty acids and glycerol. 91. Glycerol is polar and soluble in water. 92. Triglycerides contain 1 glycerol and 3 fatty acids equals 3 carbonyl esters. 93. Fatty acids equals RCOH equals 16C/palmitic acid/animals saturated and oleic acid equals 18C/plants unsaturated/2 hydrogen less per double bond. 94. Even know of carbons. 95. Triglycerides produce twice energy as compared to carbohydrates. Phospholipids. 96. 1 glycerol plus 2 fatty acids plus 1 phosphoric acid plus sometime nitrogen. 97. Phospholipid molecule is amphiphilic. 98. Glycerol head is hydrophilic. 99. Fatty acid tail is hydrophobic. Waxes. 100. Long-chain fatty acids plus long-chain alcohol equals solid at room temperature. 101. High melting point / hydrophobic / resistant to degradation / waterproof layer. 102. Reduced transpiration equals cuticle. 103. Present on leaves / sheep hairs / arthropod shells, epicuticle, lanolin, from Latin lana, wool, and oleum oil, also called wool wax or wool grease, is a wax secreted by the sebaceous glands of wool-bearing animals. Lanolin used by humans comes from domestic sheep breeds that are raised specifically for their wool. Historically, many pharmacopoeias have referred to lanolin as wool fat, adepts linnae, however, as lanolin lacks glycerides, glycerol esters, it is not a true fat. Lanolin primarily consists of sterol esters instead. Lanolin's waterproofing property aids sheep in shedding water from their coats. Certain breeds of sheep produce large amounts of lanolin. There is an inverse correlation between fiber diameter and wool wax content. Steroids. 104. No fatty acids present / 4 rings equals 17C / different functional group attached. 105. Cholesterol is steroid / equals vit D / aldosterone sex hormone precursor. 106. Aldosterone regulates sodium in blood. Terpenoids. 107. No fatty acids / isoprenoid units equals C5H8 / rubber / carotenoids. Terpenes equals terpenes / terpene / are a large and diverse class of organic compounds, produced by a variety of plants, particularly conifers, and by some insects such as termites or swallowtail butterflies, which emit terpenes from their osmeteria. They often have a strong odor and may protect the plants that produce them by deterring herbivores and by attracting predators and parasites of herbivores. 2. 3. The difference between terpenes and terpenoids is that terpenes are hydrocarbons, whereas terpenoids contain additional functional groups. They are the major components of resin, and of turpentine produced from resin. The name terpene is derived from the word turpentine. In addition to their roles as end products in many organisms, terpenes are major biosynthetic building blocks within nearly every living creature. Steroids, for example, are derivatives of the triterpene squalene. 108. Carotenoids are brown / yellow / orange / red. 109. Carotenes are orange and red / and xanthophylls are yellow. 110. The term carotene, also keratin, from the Latin carota, carrot, is used for many related unsaturated hydrocarbon substances having the formula C40HX, which are synthesized by plants but in general cannot be made by animals. 111. Beta-carotene is orange pigment which gives 2 vitamin A molecules. Function of lipids. 112. Storage molecules / seeds / fats in animals / contain more CH bonds and less oxygen than carbohydrates / lipoprotein membrane / insulation materials / 113. Plant hormones equals auxins / gibberellins / cytokinins are lipids. Nucleic acid. 114. 2 types of nucleic acids equals RNA and DNA. 115. Nucleotide equals nucleoside plus phosphate. 116. Nitrogen base is attached at C1 and pentose sugar at C5. 117. The bond between phosphoric acid and OH group of pentose sugar is ester linkage. 118. A phosphodiester bond occurs when exactly 2 of the hydroxyl groups in phosphoric acid react with hydroxyl groups on other molecules to form 2 ester bonds. An example is found in the linking of 2 pentose, 5-carbon sugar, rings to a phosphate group by strong, covalent ester bonds. Each ester bond is formed by a condensation reaction in which water is lost. This bond is a key structural feature of the backbone of DNA and RNA and links the 3' carbon of one nucleotide to the 5 carbon of another to produce the strands of DNA and RNA. 119. Purines are double-ring nitrogen base equals A plus G. 120. Pyrimidines are single-ring equals cut. 121. Nucleotides are equals mono / D / polynucleotides. 122. Adenosine equals adenine plus ribose. 123. ATP equals 7 kcal energy. 124. ATP is the energy molecule. 125, dinucleotide equals NAD/NADP/FAD. 126, coenzymes are small molecules. They cannot by themselves catalyze a reaction, but they can help enzymes to do so. In technical terms, coenzymes are organic non-protein molecules that bind with the protein molecule, apoenzyme, to form the active enzyme, holoenzyme. 127, a number of the water-soluble vitamins such as vitamins B1, B2, and B6 serve as coenzymes. 128, a dinucleotide conveying a vitamin is known as coenzyme. Polynucleotides. 129, A equals G/cytosine triple H bonds with T. 130, James Watson/Francis Crick/Maurice Wilkins equals Nobel Prize structure of DNA. 131, the diameter of DNA is 2 nanometers and base pairs are 0.34 at distance and 3.4 for DNA twist. 132, DNA backbone is sugar and phosphate. 133, 1928 Frederick Griffith equals first evidence of DNA. 134, 1944 Avery McLeod and McCarty equals experimental proof. 135, Hershey and Chase equals bacteriophage experiment. 136, DNA is the data bank of living organisms. Genetic code. 137, the information encoded in the form of a gene equals genetic code. 138, gene is the unit of heredity. 139, genes specify A, proteins B, functional RNA chains. 140, a code is a sequence of 3 nitrogenous bases, triplet, along with the sugar phosphate strand of DNA. 141, August equals methionine start codon. 142, UAA/UAG/UGA equals stop codon with exception in mitochondria UGA equals tryptophan. 143, plasma membrane equals lipoproteins/glycolipids/glycoproteins. 144, chromosome is basically nucleoprotein/slightly acidic/soluble in water. 145, glycolipids important components of brain. 146, glycoproteins present in plasma membrane. Chapter number 3. Enzymes. French chemist Anselme Payen was the first to discover an enzyme, diastase, in 1833-9. A few decades later, when studying the fermentation of sugar to alcohol by yeast, Louis Pasteur concluded that this fermentation was caused by a vital force contained within the yeast cells called ferments, which were thought to function only within living organisms. He wrote that alcoholic fermentation is an act correlated with the life and organization of the yeast cells, not with the death or putrefaction of the cells. In 1877, German physiologist Wilhelm Kuhn, 1837-1900, first used the term enzyme, which comes from Greek nuzeta upsilon mu omicron nu, leavener in yeast, to describe this process. 11. The word enzyme was used later to refer to non-living substances such as pepsin, and the word ferment was used to refer to chemical activity produced by living organisms. Eduard Buchner submitted his first paper on the study of yeast extracts in 1897. In a series of experiments at the University of Berlin, he found that sugar was fermented by yeast extracts even when there were no living yeast cells in the mixture. He named the enzyme that brought about the fermentation of sucrose zymase. In 1907, he received the Nobel Prize in Chemistry for his discovery of cell-free fermentation. Following Buchner's example, enzymes are usually named according to the reaction they carry out. The suffix ase is combined with the name of the substrate, example lactase is the enzyme that cleaves lactose, or to the type of reaction, example a DNA polymerase forms DNA polymers. Introduction. 1. Zymase equals in yeast/globular proteins. 2. Biological catalyst/specific proteins. 3. React with substrate by A, bond fit B, geometrical fit. 4. Body of enzyme equals A, framework B, binding site. 5. Types of binding sites equals A, active site B, allosteric site. 6. Active site reacts with equals A, substrate B, competitive inhibitor. 7. Allosteric site binds with equals A, non-competitive inhibitor B, feedback mechanism. 8. Active site is determined by our group of amino acids. 9. 2 to 3 amino acids are present in active site of an enzyme. 10. Holoenzyme equals apoenzyme plus cofactor. 11. Apoenzyme, an enzyme that requires a cofactor but does not have one bound. An apoenzyme is an inactive enzyme. Activation of the enzyme occurs upon binding of an organic or inorganic cofactor. 12. Holoenzyme, an apoenzyme together with its cofactor. A holoenzyme is complete and catalytically active. Most cofactors are not covalently bound but instead are tightly bound. However, organic prosthetic groups such as an iron ion or a vitamin can be covalently bound. Examples of holoenzymes include DNA polymerase and RNA polymerase, which contain multiple protein subunits. The complete complexes contain all the subunits necessary for activity. DNA polymerase uses a magnesium ion for catalytic activity. 13. First coenzyme was discovered by the English biochemist Sir Arthur Harden, 1865 to 1940, around the turn of the 19th century. Inspired by Buchner, who in 1897 had detected an active enzyme in yeast juice that he had named zymase, Harden used an extract of yeast in most of his studies. He soon discovered that even after boiling, which presumably destroyed the enzymes in yeast, such deactivated yeast could be reactivated. This finding led Harden to the realization that a yeast enzyme apparently consists of two parts, a large, molecular portion that could not survive boiling and was almost certainly a protein, and a smaller portion that had survived and was probably not a protein. Harden, who later shared the 1929 Nobel Prize in Chemistry for this research, termed the non-protein a coferment, but others began calling it a coenzyme. 14. Enzymes are switched on/off by a cofactor B, allosteric site. 15. Cofactors are equals A, prosthetic group B, metallic ions. 16. Prosthetic group. Prosthetic group is the same as the coenzyme but are tightly bound to the enzyme. When they are split off, the enzyme is mostly denatured. Examples include heme, including biotin, pyridoxal phosphate, thiamine pyrophosphate. 17. Coenzymes are F80/N80/N80P/coenzyme A, Vit B5/CoQ. 18. EF80/NAD equals respiration and N80P equals photosynthesis. 19. Prosthetic group binds with equals A, covalent bond B, H bond. 20. Metallic activators are ZN/Cu/Fe. 21. Metallic activators have ionic bonds. 22. Enzyme lower activation energy. 23. H2O2 at H2O 86 kilojoules per mole without catalase enzyme but 1 kJ/mole with catalase. 24. Many coenzymes are derived from vitamins. 25. Lack of vitamin B produce beriberi. 26. Emil Fischer 1890 proposed lock and key mechanism equals lock equals substrate and key equals enzyme. Biology first yearbook. Lock and key theory. The specific action of an enzyme with a single substrate can be explained using a lock and key analogy first postulated in 1894 by Emil Fischer. In this analogy, the lock is the enzyme and the key is the substrate. Only the correctly sized key, substrate, fits into the keyhole, active site, of the lock, enzyme. Smaller keys, larger keys, or incorrectly positioned teeth on keys, incorrectly shaped or sized substrate molecules, do not fit into the lock, enzyme. Only the correctly shaped key opens a particular lock. 27. Koechlin 1959 proposed induced fit hypothesis. 28. E plus SA E.S, transition state, I plus P. Enzymes nomenclature. 29. Oxidoreductases equals in biochemistry, an oxidoreductase is an enzyme that catalyzes the transfer of electrons from one molecule, the reductant, also called the electron donor, to another, the oxidant, also called the electron acceptor. This group of enzymes usually utilizes NADP or NAD+ as cofactors. 30. For example, an enzyme that catalyzed this reaction would be an oxidoreductase. A plus B, A plus B. In this example, A is the reductant, electron donor, and B is the oxidant, electron acceptor. 31. An oxygenase is any enzyme that oxidizes a substrate by transferring the oxygen from molecular oxygen O2, as in air, to it. E.g. Rubisco. 32. Oxidase equals final E acceptor is oxygen, e.g. NADH oxidase equals A, and A3. Temperature. 33, optimum equals humans 37C slash plants 29C slash mammalian enzymes 40C. 34, minimum equals 0C but Arctic flea equals dash 10C. 35, TAC equals 90C slash Q10 ratio. pH. 36, optimum equals 7 for most 7 to 8. 37, protease 1 pH. 38, neutral equals Cu equals urease slash catalase. 39, acidic equals PLI llama equals pepsin 1.5-1.6 slash lipase equals 4.7 slash invertase 4.5 slash lipase 4-5 slash amylase 4.6 to 5.2 slash maltase 6.1 to 6.8 slash amylase 6.4 to 7. 40, basic equals LT equals lipase, pancreas, 8 slash trypsin 8.7. Inhibitors. 41, reduced the rate of reaction. 42, used as drugs slash pesticides slash research tools. 43, competitive inhibitor is similar to substrate and compete with substrate. 44, sulfonamide is competitive inhibitor to antibacterial drug. 45, non-competitive inhibitor fit into allosteric site. 46, weak binding inhibitors are reversible inhibitors. 47, irreversible inhibitors are cyanides slash heavy metal ions and some insecticides. 48, feedback mechanism equals when the product itself bind to allosteric site and stop its formation. Chapter number 6. Prokaryotes. Introduction and taxonomy. 49, life is not possible on Earth without prokaryotes. 50, Five Kingdom system was proposed by Robert Whittaker. 51, once all prokaryotes were included in Kingdom Monera. 52, now prokaryotes have two domains archaea and bacteria. 53, domain is also called superkingdom. 54, prokaryotes are classified on the biochemical and metabolic characters. Phylogeny. 55, most of prokaryotes cannot be cultured in laboratory. 56, only 6,300 prokaryotes are given scientific names. 57, Dr. Carl Woese introduced 6 kingdoms and 3 domains. 58, both archaea and bacteria originated from Leuca. Archaea. 59, archaea plasma membrane is different from bacteria. Archaea contain terpenoids instead of phospholipids. 60, cell wall of bacteria is Murrian and archaea cell wall pseudomurrian. 61, archaea contain no peptidoglycan. 62, the prokaryotes are differentiated by rRNA-16S of ribosomes. 63, archaea are, A, extremophiles, B, methanogens. 64, extremophiles are, A, extreme halophiles, B, extreme thermophiles. 65, extreme halophiles lives in Utah's Lake and Dead Sea. 66, extreme halophiles cannot live in non-saline environment. 67, thermophiles can tolerate 90-121C0 and in Pacific Ocean 150 feet below. 68, methanogens are anaerobic use CO2 and release methane. 69, methanogens lives in A, marshes, B, animals, C, lac bottom, D, thick ice layers. Bacteria classification. 70, first seen by van Leeuwenhoek in 1648. Alpha Proteobacteria. 71, symbiotic with roots of leguminous species. 72, Rhizobium is alpha Proteobacteria. 73, fix atmospheric nitrogen. Beta Proteobacteria. 74, involved in nitrogen recycling. 75, oxidize ammonium produce nitrite. Gamma Proteobacteria. 76, photosynthetic. 77, sulfur bacteria H2S. Delta Proteobacteria. 78, myxospores, myxobacteria. 79, slime bacteria. Epsilon Proteobacteria. 80, human and animals pathogens. Chlamydia. 81, gram-negative. Spirochetes. 82, free living. 83, move by rotating internal filament. 84, never form colony. 85, notorious parasites of human syphilis caused by spirochetes. Gram-positive. 86, both solitary and colonial forms. 87, actinomycetes, phylum, the subgroup gram-positive contain Mycobacterium, 150 spp, cause tuberculosis and leprosy. 88, most actinomycetes are free-living and decomposers. 89, 2/3 of antibiotics are produced by actinomycetes. E.g., many species of actinomycetes produce antimicrobial compounds under certain conditions and growth media. Streptomycin, actinomycin, and streptothricin are all medically important antibiotics isolated from actinomycetes bacteria. 90, streptomycin is obtained from Streptomyces. 91, tuberculosis, Mycobacterium tuberculosis, and leprosy, Mycobacterium leprae. 92, mycoplasma is gram-positive without cell wall. Cyanobacteria. 93, 1 to 21%. 94, increased ozone layer. 95, oxygen-producing prokaryotes. 96, ancestor of chloroplasts. Capsule. 97, tight protective coat. 98, gelatinous structure made of polysaccharides and proteins. 99, less tight bound capsule is known as glycocalyx or slime. Structure of bacteria's cell wall. 100, muriin equals sugars plus amino acids equals 2013/2014. 101, 60 to 80% peptidoglycan equals carbohydrates protein complex. 102, antibiotic react with bacterial cell wall. 103, antibiotic cannot bind to mycoplasma equals no cell wall. 104, gram-negative bacteria contain less peptidoglycan. 105, gram-negative lose the dye. 106, EVA-LPS blocks the entry of antibiotics, therefore more resistance. Flagella. 107, Atricus no flagellae.g Streptococcus 2005. 108, Monotricus equals 1 flagellum. 109, Lophotricus equals a group of 2 or more flagella inserted at one pole of cell. 110, Amphitricus equals a group of flagella inserted at both ends of cell. 111, Poritricus equals flagella are dispersed on entire surface of cell equals 2013. 112, flagellum move like propeller. 113, flagella parts basal body / hook / filament. Pili. 114, pili on entire surface play no part in movement. 115, pilin proteins equals 6 nanometers. 116, adhesion and conjugation. 117, movement produced by type 4 pili is typically jerky, so it is called twitching motility, as distinct from other forms of bacterial motility, such as that produced by flagella equals federal board. 118, F. pilus of Escherichia coli, encoded by the F plasmid or fertility factor. 119, mutant bacteria that lack fimbriae cannot adhere to their usual target surfaces, thus cannot cause diseases. 120, during infection or to initiate formation of a biofilm equals fimbria. Cell membrane. 121, like eukaryotic cells but lipids are different equals carbohydrates and sterol absent. Sterols, also known as steroid alcohols, are a subgroup of the steroids and an important class of organic molecules. They occur naturally in plants, animals, and fungi, with the most familiar type of animal sterol being cholesterol. Cholesterol is vital to animal cell membrane structure and function as a precursor to fat-soluble vitamins and steroid hormones. 122, cell membrane resemble mitochondria of eukaryotic cells. 123, DNA replication mesosome formation mesosome cell division. 124, more prominent in gram-negative bacteria, agree to disagree. 125, cytoplasm cytosol. 126, nucleoid equals incipient nucleus. 127, 70S equals ribosomes. Genomic organization. 128, single / haploid / circular / 129, plasmid equals small circular extra chromosomal piece of DNA. 130, no gametes formation. 131, fewer up to 100 genes. 132, 1-15 / cell and antibiotic genes present. Size and shape of bacteria. 133, 2 to 10 m equals length and 0.2-2 m. Cocci. 134, non-motile/no flagellum. 135, mono/diplo/tetrasarcina/streptostaphylobacteria. Bacilli. 136, rods and curved rods. 137, Salmonella typhi and Clostridium tetani are bacillus. Spirilla. 138, corkscrew-shaped. 139, motile and flagella at the ends. 140, never form colonies. 141, contain axial filaments. 142, Vibrios equals comma-like / spirochetes equals flexible and Spirilla equals rigid. Endospore structure. 143, found in gram-positive E.G. Clostridium. 144, Exosporium, toxins / protein coat, chemical enzymatic resistance, cortex, thick peptidoglycan / temp resistance, core equals center of endospore / SASPs plus dipicolonic acid plus DNA plus ribosomes. 145, intestine of Egyptian mummies / 7,518 years alive in sediment of Minnesota Elk Lake. Photoautotrophs. 146, H2S bacteria / alpha proteobacteria 2015/2016. 147, chlorophyll is dispersed in enfolded region of cell membrane. 148, oxygenic photosynthetic bacteria perform photosynthesis in a similar manner to plants. They contain light harvesting pigments, absorb carbon dioxide, and release oxygen. Cyanobacteria or cyanophyta are the only form of oxygenic photosynthetic bacteria known to date. 149, anoxygenic photosynthetic bacteria consume carbon dioxide but do not release oxygen. These include green and purple bacteria. 150, decomposing bacteria are known as scavengers / recyclers. 151, pathogens parasitic bacteria which cause diseases. 152, mutualism equals both partners get benefits. E.g. Rhizobium radicicula, corrected. 153, commensalism equals one get benefits and another neither benefited nor harmed, PMI. 154, the term commensal is derived from a Latin word mensa, meaning table. Commensal organisms share their food from a common table. In the case of human commensals, the human host is the table. Corynebacteria such as Corynebacterium xerosus can establish themselves as resident commensals on human cornea. Comparison of photosynthesis in bacteria and cyanobacteria. 155, cyanobacteria are oxygenic photoautotrophs and bacteria are anoxygenic. 156, cyanobacteria use water and bacteria use H2S. 157, PSII is absent in photoautotroph bacteria. 158, only PSI is present in photoautotrophs bacteria. 159, bacteria produce ATP by cyclic electron flow, photophosphorylation. 160, bacteria produce NADPH by reverse electron transport chain. 161, two families of reaction centers in photosystems exist, type I reaction centers such as photosystem I, P700, in chloroplasts and in green sulfur bacteria, and type II reaction centers such as photosystem II, P680, in chloroplasts and in non-sulfur purple bacteria. 162, photosynthetic bacteria that cannot produce oxygen have a single photosystem called BRC, bacterial reaction center. 163, the pigments of cyanobacteria are chlorophyll of phycocyanin, allophycocyanin, and phycoerythrin. Growth in bacteria. 164, increase in number of population. 165, bacterial growth is affected by temperature / nutrients availability, pH and ionic concentration. 166, generation time of bacteria is 20 minutes. 167, the interval between two successive cell division is generation time. 168, exponential growth equals 1248163264. /no 2x after each generation time. 169, in 36 hours E. coli can cover the face of entire Earth. Phases of growth. 170, lag phase equals first few hours / accustomed to environment. 171, log phase equals active growth phase / disease symptoms appears in this phase / 2015. 172. Endotoxins are released in decline phase/2015. Reproduction in bacteria. 173. Asexual is most common equals binary fission. 174. Before division the cell membrane grows twice its size. 175. Diversity in bacteria come by mutation and genetic recombination. 176. Mutation involves the disruption of nitrogen base sequence in DNA. 177. In bacterial recombination fragment of the recipient DNA is replaced by donor DNA. 178. The recipient after receiving donor DNA is known as recombinant cell. Transformation. 179. Streptococcus pneumonia equals Atricus equals S-type and R-type. 180. 1928 F. Griffith plus Avery and co-worker equals 1944. Transduction. 181. Letterberg and Zinder equals 1952. 182. Bacteriophage is a third party. 183. Generalized and specialized transduction. 184. rDNA, prophage, equals donor DNA plus recipient DNA. 185. Donor DNA plus phage DNA equals prophage. Conjugation. 186. Letterberg and Tatum 1946 in E. coli. 187. Mutant 1 unable to synthesis biotin and methionine. 188. Mutant 2 could not synthesis threonine and leucine. Importance of bacteria. 189. Golden rice equals transgenic rice plus beta-carotene equals vitamin A equals prevent blindness. 190. Bioremediation equals to remove pollutants. 191. Ethanol to replace fossil fuel. 192. Gut bacteria and human microflora are commensals. 193. Fibers Redding. 194. Airborne diseases are equals diphtheria/tuberculosis/pneumonia/whooping cough. 195. Waterborne diseases equals dysentery/gastroenteritis/typhoid/cholera. 196. Foodborne diseases equals botulism plus erysipelas equals skin disease. 197. Cholera equals Vibrio cholera equals gram-negative/toxin/tetracycline. 198. Typhoid equals gram-native/5F/Salmonella typhi/chloramphenicol. 199. TB/INH/rifampin/BCG. 200. Pneumonia equals 80% streptococcus/80 strain vaccines for 23/penicillin/chloramphenicol/tetracycline. 201. Plant diseases equals XPAR equals BSRGW. Xanthomatus equals blight/pseudomonate equals spot, leaf, Erwinia equals rot/. Agrobacterium equals galls/ralstonia equals wilt. Bacterial flora of human. 202. Oral cavity equals streptococci/lactobacilli. 203. Respiratory tract equals streptococci/coronibacteria/Neisseria. 204. Urinogenital tract equals streptococci/lactobacilli/coronibacteria. 205. Large intestine equals helicobacter/bifidobacteria/bacteroids lab/methanogens/clostridia. Control of harmful bacteria. 206. 100 C0 for 10 minutes kill vegetative forms of bacteria. 207. Milk is exposed for 72 C0 for 15 seconds equals 2014 and 2005. 208. Ultra-high temperature equals 140 C0 for 3 seconds. 209. 170 C0 for 2 hours equals sterilization. 210. Bacteriostatic effect equals refrigeration from 0-7 C0 cannot produce toxins. 211. Radiation equals ionizing radiation/UV radiation/microwave radiation. 212. Phenolics equals to control gram-positive in nurseries. 213. Cl to disinfect water. 214. Disinfectants equals copper selenium/mercury/zinc equals shampoos/fish tank. 215. Glutaraldehyde is less irritating than formalin. 216. Ethylene oxide kills all germ and endospore if exposed 4 to 18 hours. Previous ETEA questions from Chapter 1. 8. The interval between two successive division of bacteria is called equals 2015/111. B. Ecological time. B. Population time. Ec. Growth time. D. Generation time. 9. Most disease symptoms appear during equals 2015/112. A. Lag phase. B. Generation time. C. Decline phase. D. Log phase. 10. Endotoxins are released only when bacteria equals 2015/113. B. Excrete. B. Reproduce. C. Decline phase. D. Stop phase. 11. Murine cell wall is composed of 2014/88. B. Calcium pectate. B. Glycolipid. C. Glycoprotein. D. Sugars and amino acids. 12. The bacteria maintain their survival by the formation of equals 2013/131. B. Hermagonia. B. Echinite. C. Zygospore. D. Endospore. 13. Bacteria living in the gut formed the association of equals 2015/43. B. Mutualism. B. Commensalism. C. Predation. D. Parasitism. 14. Milk sugar is pasteurized by heating for 15 seconds at the temperature of equals 2014/58-2005. B. 60 cB, 71 cC, 50 C. D. 80 C. 15. When the entire body covered by flagella, such bacterium is called equals 2013/167. A. Atricus. B. Lophotrichus. C. Amphitrichus. D. Peritrichus. 16. Archaea living in extreme condition is called equals 2016/124. A. Extremophile. B. Extremophyte. C. Methanoges. D. Extremogens. 17. The sulfur H2S bacteria is called equals 2016/155. C. Alpha proteobacteria. B. Beta proteobacteria. C. Gamma proteobacteria. D. Epsilon. Chapter number 10. Forms and functions in plants. Introduction. 1. Soil supply mineral elements. 2. Plants can be grown in absence of soil if fertilizer and minerals are given. 3. Mineral nutrition is the study of how plants get minerals from soil/air/water. Plant nutrition. Macronutrients. 4. 2 types. A. Primary nutrients. B. Secondary nutrients. 5. Primary nutrients are NPK. 6. Secondary nutrients are NGSCA. 7. Carnivorous, parasitic, plants get nitrogen from insects. 8. CO2 and O2 from air. 9. Hydrogen from water. 10. Lime is added to soil to make it less acidic equals 6 to 6.5. 11. There's 6 macronutrients. Micronutrients. 12. The micronutrients are 9. 13. Nzin/B/Cl/Mnco/Fe/Na/Mo/Cu. 14. Chlorosis may result if NPK and Ng is not present. 15. Necrosis is death of old tissue. 16. Nitrogen fixation MO/Co/Fe. 17. Chlorophyll requires Fe/Cl/Mg/S/N. 18. C4 photosynthesis require Na. 19. These are activators of certain enzymes Mn/Zn/Cu. 20. Ca is needed in cell wall/enzymes cofactor/cell permeability. Special mode of nutrition in plants, 600 spp. 21. 2 types. A. Active traps. B. Passive traps. Active traps. 22. Active traps equals Venus flytrap/flypaper traps/adhesive traps equals sundew/butterworts. 23. Sundew equals Drosera and butterworts equals Pinguicula. 24. Sundew equals sticky gland-tipped hairs and butterworts equals sticky layer of mucilage. Passive traps. 25. Pitfall traps equals pitcher plant. 26. Pitcher plants equals Darlingtonia/Sarracenia/Nepenthes equals all pitfall traps. 27. Vase-like modified leaf/downward pointing hairs/digestive enzymes, trypsin, in bottom. Role of stomata in gaseous exchange. 28. In dicot guard cells are kidney-shaped. 29. Monocot equals dumbbell-shaped. 30. The inner walls of guard cells are thick and inelastic and outer walls are thin and elastic. 31. The adjoining cell wall of 2 guard cells are not attached. 32. Subsidiary cell are epidermal cells surrounding guard cells. 33. Stomatal apparatus equals stomatal pore plus guard cells plus subsidiary cell. 34. Guard cells are unenucleated. 35. 90% transpiration occurs through guard cells. Transport in plants. Xylem. 36. Nageli introduced word xylem means wood. 37. The main sap-conducting tissue of plants, inorganic sap. 38. For components of xylem equals tracheid/vessels/fibers/parenchyma. Tracheid. 39. ADM narrow/tapering. 40. Lignin in cell wall. 41. Found in all tracheophyta equals wood formation. Vessels. 42. Wider/present only in angiosperms. 43. Hardwood is formed from vessels. Phloem. 44. Outside of xylem. 45. It forms inner bark. 46. Transport organic solutes. 47. Complex tissue. 48. 5 kinds of cells equals phloem fibers/parenchyma/companion cell/sieve tubes/pH ray cells. 49. Companion cells are present in angiosperm/albuminous in gymnosperm. 50. Phloem ray cells equals these are longitudinal parenchyma present in SSE. Phloem protect other cells. 51. Sieve tubes equals elongated living cells. 52. Sieve plates equals perforated area of end walls. 53. Companion cell equals elongated living/supply energy to sieve tubes. Water status in plants. 54. Water potential is directly related to concentration of water molecule. 55. Psi W equals Psi P plus Psi S. 56. Psi W is measured in kPa equals kilopascal. 57. Solute potential equals osmotic potential equals directly proportional to solute molecules. 58. Pressure potential equals turgor pressure developed as a result of endosmosis. 59. Pressure potential equals erectness of and shape maintenance in soft plants. Movement of water through roots. 60. Apoplast movement equals movement through cell wall/non-living pathway/intercellular. 61. Water from root hairs passes cortex/endodermis/pericycle to xylem. 62. Symplast pathway equals living pathway/cortical cell by osmosis. 63. In symplast pathway both cell membrane plus cytoplasm equals partially permeable membrane. 64. Vacuolar pathway equals attached cell have interconnected vacuoles. 65. Vacuoles are interconnected through plasmodesmata. 66. Plasmodesmata are cytoplasmic strands through adjacent cell wall. 67. Vacuolar pathway is special symplast pathway. 68. Leaf xylem and mesophyll generate main uptake power. Water movement in xylem through tact mechanism. 69. For important forces transport water solution from roots. 70. Xylem element of stem to leaves. 71. Transpiration, evaporation plus tensile, stretchy, strength of water. 72. Adhesion, the attractive forces between water molecule and cellulose. 73. Cohesion, attractive forces between water molecules/H bonds/equal to tensile strength of steel wire. 74. Tension, the pulling force/surface tension/develops in leaf spaces/negative pressure. 75. Tension equals a force that pull water from greater concentration/solar energy. Sugar starch theory. 76. Absorbing CO2 lowers H plus of cell sap equals photosynthesis 10x than respiration. 77. pH of guard cell increase/phosphorylase covert starch into glucose and phosphate equals open. 78. DPD equals diffusion pressure deficit equals sucking power/due to increase in solute concentration. 79. The amount by which diffusion pressure of a solution is lower than its pure solvent is known as diffusion pressure deficit, or DPD. This term was first coined by B.S. Meyer in the year 1938. DPD of pure water is the lowest, dilute solution is high, concentrated solution is highest. 80. DPD equals osmotic pressure, turgor pressure, water pressure. 81. During dark equals substomatal CO2 increases equals decrease in pH of guard cells. 82. Low pH phosphorylase convert glucose plus phosphate to starch equals stomata closed. During day. 83. Starch equals G6P pepapep plus CO2 malic acid malate anion plus H+. 84. H+ transported to epidermal cell and K+ ions are taken into guard cell. 85. Increase in K+ concentration in vacuole of guard cell increase DPD of guard cells equals open. During dark. 86. CO2 increases in substomatal cavity equals no H+ from guard cells equals no K+ ions into guard cells equals pH of guard cell decreases K+ is inhibited by abscisic acid. 87. In guard cells equals malate anion plus H+ equals malate equals K+ ion out of guard cell equals DPD decreases equals flaccid equals closed guard cells. Translocation of organic solutes. 88. Green leaves are source of assimilation. 89. Sucrose is transported to stem/bud/fruit/seed/root/underground stem equals sinks of assimilation. Pressure flow mechanism. 90. E-food is translocated through phloem. 91. Phloem is double circuit and xylem single circuit. 92. In early spring roots are source of assimilates. 93. Source and sinks are reversed according to season. 94. Phloem sap equals water plus sucrose, but amino acids, other sugars and hormones also transported. 95. Sugar is moved in the companion cell through active transport. 96. Sugars are taken by active transport by sink and converted to starch or cellulose. Homeostasis in plants. 97. To maintain or nearly maintain constant internal environment. 98. Osmoregulation, control of gain or loss of water. 99. Isotonic equals no diffusion / hypotonic equals endosmosis / hypertonic equals exosmosis. Hydrophytes. 100. Hydrilla / vallisneria / potamogeton equals root hair absent / whole surface absorption. 101. Large leaves / stomata on upper surface. Mesophytes. 102. Citrus / brassica / pea / peach / rose. 103. Stomata on both sides. Xerophytes. 104. Cactus / opuscia / aloe / acacia / ruscus / calotropis / ziziphus. 105. Deep roots / leaves modified into spine / hairy stem / thick cuticle / few stomata and sunken. G. pinus. Halophytes. 106. Mangrove / salt tolerator / xerophytic character / water storage tissue. 107. Salsola / rhizophora. Low temperature. 108. Thick bark / short life cycle equals ephemerals plants / composition of solute changes / hard leaves and stem / scale leaves / oils and sap / e.g. Arabidopsis. High temperature. 109. Extensive branches of roots / thick cuticle / sunken stomata / succulent / support in plants. 110. Herbaceous plants are known for their attractive flower and foliage. 111. Non-woody plants are turgid because of parenchyma cell. 112. The loss of water result in wilting. 113. The pressure of the cell pushing against the wall is turgid pressure. 114. Calenchymma means glue cells. Calenchymma. 115. Found in leaf and elongated stem. 116. Found in petiole / peduncle / pedicel / leaf margins. 117. The hollow cylinder around the vascular tissue. 118. Uneven thickened walls. 119. For types angular lamellar lacunar. 120. No lignin is found. 121. Transitional cells are present in between calenchymma and parenchyma. 122. Can do meristematic activities / living. Sclerenchyma. 123. Hard cell / non-living / strength and support. 124. Present in parts which have ceased elongation. 125. 2 groups A, fibers B, scleriids equals cellulose plus lignin. Fibers. 126. Fibers are present in flax / hemp / jute / ramii. 127, fibers are produced by cambium and procambium. 128, always lignified. 129, extraxillary fibers are known as bast fibers equals phloem fibers. 130, U fibers are also known as prosenkymatous cells. 131, associated with xylem. 132, ropes/fabrics/mattresses equals fibers. 133, flax equals Linum usitatissimum equals 3000 years equals hemp equals cannabis sativa. 134, jute equals Quercus capsularis plus rami equals Beimeria nivea equals soft textile. Sclerids. 135, isodiametric/prosenkymatous/forked/branched. 136, scleriads are short equals stone cells equals pairs equals Pyrus communis. 137, shoot of wax plants/Hoya carnosa equals stone cell. 138, shells of gnus plus stones of drupes equals scleriads. Growth and development in plants. 139, increase in no and size of cells. 140, three phases in plant cell division/elongation/maturation and differentiation. 141, juvenile cells equals meristems. Meristematic tissue. 142, apical plus lateral equals position meristem. 143, apical meristem give rise to equals protoderm/ground meristem/procambium. 144, the epidermis is formed from protoderm equals lenticel/stomata/waxy surface. 145, cortex is formed from ground meristem. 146, procambium form equals primary vascular tissue/primary phloem/primary xylem forward/vascular cambium. 147, the innermost tissue is pith equals produced by central ground meristem. 148, pith function for a short period as storage tissue. 149, apical meristem increase length of plants. 150, the elongation equals primary growth. 151, all vascular plants equals primary growth. Lateral meristem. 152, increase thickness. 153, two types A, vascular cambium B, cork cambium. 154, secondary vascular tissues are formed by vascular cambium. 155, cork cambium equals cork cells equals protect from water loss/pathogens. 156, procambium also forms secondary cambium. Growth correlation. 157, development of every organ is affected by some physiological process occurring in another organ. 158, vegetative growth is affected by fruiting. 159, the process in leaves control flowers and floral buds. 160, inhibitory correlation equals apical bud inhibit lateral buds. 161, complete equals sunflower and incomplete equals tomato. Annual rings. 162, dark late wood plus early pale wood. 163, the outer bark is formed of phloem equals retidome. 164, xylem becomes the wood. 165, vessel wood equals dark wood equals late summer wood. 166, early spring wood equals sap wood equals living cells wood. Growth responses in plants. Auxins. 167, produce an apical meristem of coleoptile. 168, IAA/control of shoot length equals cell elongation. 169, relaxation of cellulose fibrils. 170, vascular cambium differentiation and cell division in V.cambium. 171, apical dominance plus inhibit growth in main root system. Gibberellin. 172, discovered in parasitic fungus. 173, bolting in dwarf plants. 174, foolish seedlings internode elongation. 175, cause the alurone layer of the endosperm to produce alpha amylase. 176, alpha amylase breakdown starch to sugars. 177, break dormancy of seeds and stop inhibitory substances. 178, promote flowering/more fruits/seedless grapes/improve banana storage life. Cytokinin. 179, promote cell division and bud formation/stimulate protein synthesis. Abscisic acid. 180, abscission 2 equals bichulate and cotton plants equals dormant equals bud dormancy. 181, inhibit active growth of seedling/flowering in long day plants. 182, stomata closing/stop uptake of K+ by guard cells. 183, facilitate influx of water into roots equals also called stress hormone. Ethylene. 184, gaseous hormone/inhibit auxiliary bud/stimulate fruit ripening. 185, decrease in auxins increases ethylene equals senescence. Plant movements. 186, turgor movements equals reversible movements equals gain or loss of water. 187, turgor movements equals bulliform cells equals longitudinal row in epidermis. 188, pulvinus equals dropping of leaves equals bases of leaves. 189, pulvinus equals parenchyma cells with large intercellular spaces plus VB. 190, unequal movement of water in cells. 191, turgor movements equals A, rapid movement B, sleep movement. Rapid movement. 192, mimosa podaca. Sleep movements. 193, leguminosae equals pulvinus equals sleep movements. Growth movements. 194, self-controlled or induced by external stimulus. 195, irreversible movements equals autonomic and paratonic. Autonomic movements. 196, shift as whole equals chlamydomonas/volvox/zoospores/motile gametes/. 197, gametes moved to stimulus i.e. sweet fluid/cyclosis is autonomic movement. 198, 3 types, A. Epinasty B, hypinasty C, nutation. 199, epinasty equals opening of bud equals upper growth is more. 200, hypinasty equals keep the bud closed equals lower surface more growth. 201, nutation equals zigzag movements equals one side more growth equals Japanese not weed. Paratonic movements. 202, 3 types tropic/nastic/tactic. Tropic movements. 203, partial directional movements/organ movements. 204, hydrotropism equals moisture is stimulus equals roots/rhizoids/hyphae. Thigmotropism equals stimulus of touch/tendrils/passiflora/lathyris/smilax equals positive root also show negative thigmotropism equals stamens dehiscence. 205, chemotropism equals pollen tube to egg due to disintegration of synergid cells equals Drosera, sundew. Tentacles show positive chemotropism equals chemicals like proteins/phosphates and salts of ammonia cause these tentacles to bend/hyphae of fungi positive chemotropism to sugar/peptones and negative to acids/alkalis. Gnostic movements. 206, 3 types photonasty/thermonasty/seismonasty. Photonasty. 207, light/oxalis flower close during night/pulvinus/. Thermonasty. 208, crocus/tulip/flowers responses due to temperature variation. Seismonasty. 209, touch or fraction/shock movements/pulvinus/stimulus is external. Tactic movements. Phototactic. 210, whole organism moved to light. 211, chlamydomonas/zoospores of Eulithrix/volvox/chloroplast. 212, thermotactic equals unicellular algae. 213, chemotactic equals gametes/spermatozoids of bryophytes and pteridophytes move to sugars and proteins towards archegonia. Photoperiodism. 214, photoperiod equals relative length of day and night. 215, photoperiodism equals response to photoperiods. 216, short day plants early spring plants equals tobacco/dahlia/soya bean/chrysanthemum equals CV equals max. 217, long day plants equals summer plants equals hibiscus/beet/spinach/potato equals CV equals minimum. 218, day neutral plants equals independent of day length equals maize tomato sunflower cucumber. Phytochrome. 219, present in the tissue of higher plants. 220, sensitive to light transition. 221, 2 forms PR and PFR. PR equals 660 and PFR equals 730. PFR is active form. 222, PFR promote flowering in long day plants and inhibit flowering in short day plants. 223, PFR equals changes in seedling and anthocyanin development. Flowering hormone/florigen. 224, MH Chalicane 1936. 225, Florigen is produced in leaves/chrysanthemum equals short day plant. Vernalization. 226, the conversion of winter variety into spring variety. 227, vernalin hormone equals early development of flower equals ripening of crops/4C effective. 228, biennial/perennial plants are stimulated to flower. 229, stimulus is received by embryo/stem apex/Siberia/1930/1940/Lysenko 1928. 230.